Hi, my name is Liangen Yu, a PhD student in Animal and Food Science Department. Today, I would like to talk about the effect of the delayed feeding, a kind of early life stress, on intestinal epithelial stem cells in broiler chickens. So broiler chickens are 21.7 billion volumes in United States. Delayed feeding is commonly present in chickens industry due to maximal efficiency. However, previous studies indicate delayed feeding in chickens has negative impact on gut health and performance, such as lower body weight, in both early and later life in chickens. But how the early life stress affects gut health is unclear. Since the delayed feeding affects both early and later life in chickens, it might be associated with the intestinal epithelial stem cells, which differentiation into epitheliums to maintain a normal intestinal function. Here we hypothesize that delayed feeding affects intestinal epithelial stem cells, alters intestinal epithelium population, and impacts on the gut health. We use 72-hour delayed feeding chickens as our models. We took samples on day 3, day 14, and day 21 to represent the effect of delayed feeding on immediate stress, early and later life in chickens. So let's move to the result part. First, we measure the intestinal morphology with HE staining and intestinal permeability with using chamber to represent the gut health. Delayed feeding decreased the villus height, cryptid, and cryptfission in day 3 and day 21 chickens. Intestinal permeability increased in day 3, day 14, and day 21 delayed feeding chickens. The data suggested 72-hour delayed feeding decreased the gut health in chickens. So next, we identify the population of intestinal epithelium by real-time PCR. So the chicken, mucin2, and LYZ represent the endoendocrine cell, goblet cells, and penny cells, respectively. PCNA represents the proliferation, and both LGR5 and OFN4 represent the intestinal epithelial stem cells. So the nutrient transporters were measured with Western blot to represent the enterocyte also. In our results, we can see that the delayed feeding altered population of intestinal epithelium, especially endoendocrine cell, penny cells, and intestinal epithelial stem cells. We further stain the specific epithelium to confirm the number and location of penny cells and endoendocrine cells. We found that the total number of penny cells and endoendocrine cells match the real-time PCR data, which I told you previously. However, the location is different. The difference number of penny cells in day 14 delayed feeding chickens majority comes from the villus area but not the crypt area. In endoendocrine cells, the increased number of cells majority comes from the crypt and lower villi area. But in day 14 chickens, the higher villi area decreased in delayed feeding chickens. So another published paper indicates that the location difference of endoendocrine cells express different endoendocrine. In other words, the results suggest that the different location of endoendocrine cells and penny cells might have functional change. In the final, we use Android culture system and in vivo BRDU injection to measure the ability of intestinal epithelial stem cells. BRDU positive cells significantly decreased in delayed feeding chickens. Enteroid formation rate, enteroid size, and expansion also significantly decreased in delayed feeding chickens. Our data suggests delayed feeding decreased the intestinal epithelial stem cells ability. So in the conclusion, we confirm that the delayed feeding decreased the chicken's gut health, altered the population of intestinal epithelium, and decrease the intestinal epithelial stem cells ability. The relationship between the three aspects needs to be further investigated.